Alrighty, screencast time again. This one is addressing redox reactions, that is reduction oxidation reactions, and how they relate to cell respiration and ultimately how they're going to relate to photosynthesis. So I just want to introduce you here to what an oxidation is, what a reduction is, and then sort of talk about the relationship between those two. We're gonna, if you're an AP Chem person, this is going to be a little bit of a different perspective than what you would see in chemistry. But just, don't go complaining to the AP Chem teachers that I don't know what I'm talking about. So here we go, cell respiration. We had looked at this image before, and we want to point out that in respiration, we're going to be taking organic molecules like glucose, right? And we are going to be, which is C6H12O6, and what we're going to be doing to them is oxidizing them. And we're going to oxidize them completely to CO2. And in the process, we're also going to make a water molecule. And since this is aerobic respiration, this is where the oxidation part comes in, is that this oxygen is going to play a key role as well. So again, we're going to be taking organics, and in the presence of oxygen, we're going to be oxidizing them to CO2. Right? And along the way, we're going to be then reducing something as well. So and one more thing before I leave this slide, we're going to be talking also about these NADH and FADH2 molecules along the way here today a little bit. So oxidations and reductions. Oxidations is when you are losing, and reduction is when you are gaining. And the question is, well, losing or gaining what? And the answer is electrons. Right? So electrons, right, a loss of electrons is oxidation, and a gaining of electrons is called a reduction. Now, for biologists here, we're dealing primarily with organic molecules, carbon, hydrogen, and oxygen. And an easy way to kind of keep track of which molecule is being oxidized and which one's being reduced is to sort of follow the hydrogens. Again, because a hydrogen is one proton and one electron. So if something loses a hydrogen, if an organic molecule loses a hydrogen, it's losing an electron as well. So if we can kind of keep track of where the hydrogens go, then that is an easy way to sort of know which one is being oxidized, which one is being reduced. So let me just go back over here a second. Why the picture of the lion and why the picture of the oil rig? Because lion here is a Leo, right? Leo is lion, and this one is Goering, right? Here he is, Goering. Grr, Leo the lion says Grr, and this is the way to remember which one is which. Losing electrons is oxidation, gaining electrons, reduction. So again, losing electrons, oxidation, gaining electrons, reduction. It also works down here with an oil rig. This is an oil rig. Oxidation is losing electrons, reduction is gaining electrons. Again, oxidation is losing electrons, reduction is gaining electrons. Okay, so let's put this into perspective. So before we move on here, the relationship is that these two things are inverses of one another. So the relationship, if some molecule is oxidized, another molecule is reduced. And these things go hand in hand. Molecule is reduced. So if something is going to be oxidized, something else has got to be reduced, and they go hand in hand. You can't have an oxidation without a reduction. You cannot have an oxidation without a reduction, and vice versa. You can't reduce something without something else being oxidized. So in terms of respiration, what's that look like? Again, here we go. We're going to have this organic molecule over here, and this is an example is a carbohydrate, glucose, and we're going to have then oxygen. So when we talk about aerobic respiration, it's a series of redox reactions. The whole overall thing is that this organic molecule is going to be oxidized. So again, if I'm going to oxidize this organic molecule, I'm going to remove electrons from it. All right, electrons are going to be lost from it. And so we're going to lose electrons. And this means that we're going to lose hydrogens. And so if I take hydrogens from this organic molecule, from an organic molecule, right, what am I left with? Well, if I take the H away, I'm left with just CO, which is carbon dioxide. So this is how we say this. We say that the organic molecules are going to be oxidized to CO2. This glucose is oxidized to carbon dioxide. This is how we describe this. So in respiration, organic molecules are oxidized to carbon dioxide. Well, I just got done saying on the previous slide that you can't oxidize something without reducing something else. So the question is, all right, well, what's going to be reduced? Well, I took the hydrogens away from the CHO, and where am I going to put them? I'm going to put them onto this oxygen molecule. So oxygen, we say, is going to be reduced to water. And oxygen is reduced to water. Well, if you notice, this makes sense to you, right? Because the CHO, I lose an H, I just have CO. And if I have O and I add an H, then I get H and O. Now the stoichiometry all works out here, and I'm not worried about that at the moment. So aerobic respiration is ultimately a redox reaction, but it doesn't just happen in one step. There are lots and lots of steps that go on there. Okay, so take a minute and summarize down here. Identify which molecule was oxidized, and then identify for yourself which molecules were reduced. If you're a little bit really understanding this, you're talking about this reducing agent and then the oxidizing agent. This is just kind of extra down here, but the reducing agent would be the thing that is doing the reducing, and then the oxidizing agent is the thing that is doing the oxidizing. So if you're a reducer, you're giving electrons, you're giving hydrogens to something. If you're an oxidizing agent, then you're oxidizing something. That means you're pulling electrons away from a molecule. So it might be a good idea to pause here and sort of answer those two questions, or those four if you're trying there. So I'll wait. Okay, if you are coming back from it, pausing that, identify which molecule was oxidized. You would say that the C6H12O6 was oxidized, and which molecule was reduced? Well, the oxygen was reduced. And when you oxidize this glucose over here, you would end up getting CO2. When you reduce oxygen, then you end up with water. So the reducing agent, the thing that was giving up giving up its electrons, this is the same answer, C6H12O6, and the oxidizing agent is oxygen. Right? Well, okay, oxidizing agent, this is the thing that's pulling electrons. Oxygen is extremely electronegative. So since it's electronegative, it's going to pull electrons, right? Oxidize things, rip hydrogens, rip electrons off of things. So oxygen is a good oxidizer. Go figure. NAD, NADH here. So if we look at this, we're going to see this several times, but let's just talk about this in terms of redox reactions. These NADH molecules are intermediate energy carriers. This is how they're referred to as in respiration circles. So this whole thing on the left over here, this is NAD+. Now you do not have to, please do not memorize the structure of this thing at all. But what I want to call your attention to is the fact that this is NAD+, right? So if it's plus, this means that it is electron deficient. Electron deficient, it doesn't have, it's missing electrons. Missing electrons. So we can give electrons to this NAD+, and if we give electrons to this NAD+, in biology and organic systems here, when we give electrons, we actually do it in the form of giving it a hydrogen, right? So if we take NAD+ and add hydrogens to it, we are reducing, so if I'm going this way, we say that NAD+ is reduced to NADH. NADH, this is now NADH, this is indicating here that it has been reduced. So this is the reduced form of NAD+. plus. We could then also describe NAD plus as the, well, anyone? We could describe NAD plus as the oxidized form of NADH. Right? So if we're going to go this way, we're reducing NAD plus to NADH, and if we go back this way, then we are oxidizing NADH. So NADH is, if we're going this way, is oxidized to NAD plus. And you're giving up hydrogens here. If you go this way, you're giving up hydrogens going this way. This will sort of, we're going to talk about this when we do the details of the electron transport chain a little bit later. Okay. Speaking of electron transport chain, what's going on in this series of reactions is if we look at the left side of this here, if we were just to add hydrogen and oxygen together to get this, this is a, negative delta G reaction here, right? So explosive loss of this heat energy. And this is not what's happening inside the cell, though. So what's happening inside the cell is on this side of the diagram. And what we're going to do is we're going to take hydrogens from food via NADH, right? From food, meaning that, read food here, read organics. I'm going to take those hydrogens from organics, and we're going to put them on NAD plus, and we're going to take them, when we put them on NAD plus, it's going to be NADH, and then those hydrogens, as we pull those hydrogens off, we're going to donate the electrons to an electron transport chain. And as the electrons move through the electron transport chain, we're then going to be able to use this negative delta G. We're going to harness this negative delta G of these electrons moving to then generate ATP, which is a positive delta G. So we're going to couple this negative delta G reaction to this positive delta G reaction here in the electron transport chain. At the end of the chain, the electrons will be donated to this oxygen atom here, and we're going to make water down here. So we're going to reduce oxygen to water at the end of that electron transport chain. But it doesn't just happen in one step. We have this multiple step process here. So, but again, oxidizing up here and reducing down here. So we're going to be reducing oxygen. What are we then going to be oxidizing? We're going to be oxidizing NADH. Going back a step, we're going to be actually oxidizing organics and reducing NAD plus to get this NADH. So if we do go back to here, and let's end where we started. So C6H12O6 organics, right? We're going to have NAD plus here. So we're going to oxidize this glucose. We're going to reduce this NAD plus. When we reduce NAD plus, we get NADH. When we oxidize glucose, ultimately over here, we're going to be getting CO2 and CO2. Along the way, we're going to be oxidizing some intermediate organic molecules here, right, to be reducing NAD plus and FAD, reducing those things to NADH and FADH2. And then over here in the electron transport chain, we're going to be oxidizing NADH and FADH2, right? And we're going to be reducing oxygen, right, to water. And in that process, we make a lot of ATP down here. How or why, that's a whole other lecture. So reductions, oxidations, moving electrons from one place to another, right? And knowing what's oxidized, what's reduced is important. Okay, there's probably a bunch of questions and confusions about that, so bring them to class and we'll work on it then. Thanks.